All right, this is page M3. Let's start here. Here we have a sperm, a human sperm and a human egg. Now, we learned last time that a human sperm or a human egg by themselves, a sperm alone or an egg alone, cannot grow into a person. Why not? Because a sperm and an egg each possess only the haploid number of chromosomes. Each one has 23, which is only half the necessary amount of genetic information required in order for a cell to grow into a person. Sperm and eggs are known as sex cells or gametes. And we'll see these words are written on page M1. When the sperm unites with the egg, and that's called fertilization, that occurs normally, we know, in the fallopian tubes, and it results in the formation of what's called a zygote. A zygote, or fertilized egg, which has now 23 plus 23, or 46 chromosomes. That's known as the double number, or diploid number. And that cell has the potential to grow into a whole new person. In fact, this cell is the first body cell of a new person. Now, a body cell is more properly called somatic cell. Somatic. S-O-M-A-T-I-C. Soma means body. So we'll see all these words written on page M1 more neatly. So this body cell, this first body cell of this new person, will then start to undergo division, and it will divide over and over and over again, increasing in numbers of body cells. Eventually, as an adult, you were made up of about 60 trillion somatic cells, or body cells. All of the cells in your body are actually all genetically identical to one another, because every body cell that develops from this zygote is genetically, in a sense, a clone of it. And so this concept of cloning, this is a natural clone. This is simply making more body cells that have a copy of whatever those original 46 chromosomes were in the zygote. They are replicated or duplicated in the nucleus of every body cell that developed from that zygote. So every cell in your body, even though they don't look the same, even though a human skin cell looks different from a human pancreas cell, which looks different from a human brain cell, in each cell there's a nucleus. And in that nucleus are a copy of those original 46 chromosomes that were in the very first cell you started out as a zygote. Okay, now, so what is mitosis? Mitosis is the name of this process by which we make more genetically identical body cells. We're going to see that written in just a moment. Now, we mentioned last class meeting, and we'll develop this, that of the 23 so-called chromosomes that are in the egg, 22 of them are called autosomes, and one of them is an X-sex chromosome. That's the chromosomes that are in human eggs. Now, what is an autosome? Autosomes are chromosomes that determine physical characteristics, such as eye color, such as blood type, such as whether you have freckles or dimples, how tall you are, your physical characteristics. That genetic information is on the so-called autosomes. Anybody know what auto means? Self. Now, the 23rd chromosome is called an X-sex chromosome. And one of the things it determines is gender, what gender you are. Now, in the sperm, and we covered this last time, the sperm, similarly to the egg, has 23 chromosomes. 22 of them are autosomes, just like in the egg. The 23rd is a sex chromosome. But in the sperm, half of the sperm produced by a guy contain an X-sex chromosome, and half of them have a Y. I want to emphasize, sperm doesn't have both. It's either one or the other. 50% of the sperm have the X, 50% have the Y. So we learned last time that if the sperm has an X and it unites with an egg, which always normally has an X, So that means that in this zygote, you would end up with 2 X sex chromosomes. And that's a genetic female. We talked about that last time on page M1. On the other hand, if the sperm was carrying the Y sex chromosome instead of the X, and it unites with the egg, which always normally has an X, then the zygote would have 1 X and 1 Y. The X from the mother, the egg from the mother, the Y from the sperm from the father. That determines a genetic male, a male. So in this, in fact, we wrote that in the zygote, yes, there's 23 plus 23 or 46 chromosomes, but in fact, analyzing this a little bit better, there's really 22 autosomes in the egg and 22 autosomes in the sperm. So what we actually have are 44 autosomes, or better yet, 22 pairs of autosomes. And we have 1 pair of sex chromosomes, either an X and an X or an X and a Y. So that's what we have. So there are 22 so-called matching pairs of autosomes and 1 pair of sex chromosomes. Now we can go and take any cell from a person's body. We can take, because they're all genetically the same, so we can take a skin cell, a liver cell, a white blood cell, any cell from your body or even a fetus. And we can analyze what chromosomes are in the nucleus. When we do that, that's called a karyotype. The word karyotype means chromosome analysis. Now that's not really such a new word, even for us in lecture. For people in lab, they've already learned this. Because in lecture, we have talked about prokaryotic cells and eukaryotic cells.
You'd say, when did we ever learn that?
At the beginning of our discussion on cytology or cells, section E. So on page E1, we learned about a prokaryotic cell, which is a cell without a nucleus, and a eukaryotic cell, a cell with a nucleus. Page E1. So karyo means nucleus. So when we analyze the nucleus, we're really analyzing what's in the nucleus, which are the chromosomes. So that's really a chromosome analysis, or nucleus, typing the nucleus. So when we do it, what we normally see is something like this. This shows the 22 pairs of autosomes. Pair number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22 pairs of autosomes. And then there's a pair of sex chromosomes. And it's either 2 X's or 1 large X and 1 itty-bitty Y. And that determines whether you're XX or XY. That determines whether you're a genetic male or genetic female. So this is what we would see in a normal chromosome analysis of a normal human cell. Now, sometimes when we analyze the chromosomes in a human cell, we don't see the right number of chromosomes. And when that happens, as we'll see, that's known as a chromosome anomaly. Before I go any further, let me just show you one more thing on this diagram. We have mentioned that the name of the process of making more body cells or somatic cells is called mitosis. Mitosis is making more somatic cells or body cells. That's how we increase and grow in size. There is another type of cell division called meiosis. Yes, it's unfortunate that the word meiosis sounds a lot like mitosis. They both start with the letter M. I didn't invent these words. But meiosis is the name of the special type of process that just occurs in our reproductive organs. That's the process of making sex cells. And where we make sex cells, where eggs are made in a woman is in her ovaries, and where sperm are made in a guy are in his testes. And this production of sex cells is called meiosis. Now, let's summarize some of the concepts that we've spoken of so far. Let's go back to page M1. So on page M1, so on the top of M1, we wrote sex cells and somatic cells. Sex cells are also called gametes or germ cells. Sometimes people wonder, why do we call them germ cells? Do they have bugs? Do they have germs? We're using the word germ like when you plant a seed in the ground, we say it germinates or grows. So these sex cells, when they unite, they can grow into a person when they unite. Anyhow, we'll call them sex cells or gametes. That's a term we'll emphasize. Anyhow, they include sperm and ova. Each possesses the haploid number of chromosomes. In humans, that's 23. So we've said of the 23 chromosomes that are in a sperm or in an egg, 22 of them are autosomes, and then there's a sex chromosome. In the egg always has an XX chromosome. The sperm contains either an X or a Y. Not one of each, but one or the other in the sperm. The body cells have XX or XY, but a sex cell has either one or the other. Each sex cell, we wrote, each sperm produced in the testes, each egg produced in the ovaries is genetically unique. So no matter how many sperm or how many eggs an individual produces, each one is a different genetic combination. Now, the formation of these sex cells occurs in our reproductive organs, and the name of the process for making them is called meiosis. We also use the terms spermatogenesis and oogenesis. We use those terms in section L. Making sperm in the testes, making ova or eggs in the ovaries. Now, what are somatic cells or body cells? We wrote that the fertilized egg or zygote is the first somatic cell of an individual. The root soma means body. Now, each body cell, each somatic cell has the double number, the diploid number of chromosomes, which in humans, that's 23 pairs or 46. There's 23 pairs, 46 chromosomes altogether. I like to say pairs. One half came from the sperm from the father, one half came from the egg or ovum from the mother. When we analyze those 23 pairs, there are 22 homologous. The word homologous means matching. 22 homologous or matching pairs of autosomes and one pair of sex chromosomes in each body cell. Now, a body cell or somatic cell with two X's is a genetic female. A body cell with an X and a Y is a genetic male. Each somatic cell that develops from the fertilized egg or zygote is genetically identical. All the body cells are genetically identical. The process of making more body cells, more somatic cells, is called mitosis. And these somatic cells undergo mitosis to grow bigger in size, because how we grow bigger is we're made up of more and more and more cells, body cells, and also for the replacement of lost or injured cells. Every time you cut yourself and some cells in your skin or anywhere else in your body die, we can make new body cells by mitosis. And that's how we make new cells. Now, let's go back to page M3 because there's one question that is commonly a question that bothers students. It's actually a very simple answer, but I'll address the question. Here's your question for many of you. Why is it that the eggs only have an XX chromosome, but we said that half the sperm have an X and half the sperm have a Y? Why can't the eggs have a Y? Why don't all the sperm have X's? Here's why. It's actually very simple. Let's say that the sperm has an X and it unites with an egg with an X, so that forms a zygote with two X's. That's a genetic female. So, if this is the first cell of a female's body, of a woman's body, remember every cell that develops from that zygote is genetically the same as that zygote. That means every cell, in the nucleus of every cell in a woman's body are two X sex chromosomes. That's what she began with, is two X's in the zygote, and there will be two X sex chromosomes in every cell of her body. So how could she possibly produce an egg containing a Y? There's not a Y sex chromosome in any cell of her body. So in her ovaries, when she makes sex cells, they have to have an X sex chromosome. There is no Y sex chromosome in any cell of her body. On the other hand, what if this sperm carrying the Y unites with the egg with the X? That means we now have a zygote with an X and Y. That's a guy. And in the nucleus of every single cell in his body is an X and a Y sex chromosome in every somatic cell or body cell. So every single cell in a guy's body, in his brain, nucleus of his brain cells, in the nucleus of his liver cells, in the nucleus of his skin cells, is an X and a Y sex chromosome. So then we can understand how in the cells of his testes, he can produce sperm, some with X's and some with Y's. Does everybody follow that? So that's how it has to work. Now, sometimes in the forming of eggs or forming of sperm in our reproductive organs, mistakes happen. And that would mean that if the egg or the sperm has the wrong number of chromosomes, that means that when the sperm and eggs unite, we're going to have a baby with the wrong number of chromosomes. This is called a chromosome anomaly. If you look on page M2, so these are called chromosome anomalies. What does the word anomaly mean? It means abnormal number of chromosomes. This results from defects in the sex cells, in the sperm or the egg. Now there are all kinds of defects that can happen. Again, when one asks the question, could this happen or could that happen, the simple answer is anything that you could imagine could go wrong, unfortunately does go wrong. But the good news is most of the time everything goes right. Now, I'm just going to talk about the most common chromosome anomaly that tends to happen. And that's something called Down syndrome. Down syndrome is an extra autosome. Basically, let me first explain. I wrote at the bottom here that we can analyze, we said, the chromosomes in the nucleus of any cell. An adult, cell from an adult, even a cell from a fetus. And when we analyze the chromosomes, that's called a karyotype, chromosome analysis. Now, let's just show you, I wrote the word amniocentesis and chorionic villus sampling. Let's look on page M6. On M6, so this...
If a baby's born with two sex organs, what's that?
We're not going to talk about it. Okay? So, on page M6, we've got enough to focus on what we need to know. All right? So, we're talking about the most common things, not the least common things. On page M6, so we can, obviously, if we wanted to analyze the makeup of chromosomes in our cells, we can just take any cell from our body, a skin cell, a white blood cell, any cell. But how would we do it from a fetus? So, an older method is known as amniocentesis. Amniocentesis is where a needle is inserted through the abdomen of the mother and into the amniotic sac surrounding the baby. We talked about how there's really two sacs surrounding the baby, an inner amniotic sac and an outer chorionic sac last time. And basically, some fluid containing some of the cells of the baby will be withdrawn into the syringe, and then we can analyze those cells and see if there's any problems with those cells. A newer method is known as chorionic villus sampling, or CVS, like the drugstore. This is where an instrument is passed up the vaginal canal or birth canal, and a small snippet of cells is snipped off or removed from that outer sac surrounding the baby. What did we call that outermost sac? The chorionic sac. So this is called chorionic villus sampling. Now you might say, well, wait a second. Why would you want to sample that sac? Aren't the sacs surrounding the baby, aren't they made from the mother? No. All the sacs, both the inner amniotic sac and the outer chorionic sac, develop from the baby. And they are made up of the baby cells. Now our first thought is, well, how could these sacs surrounding the baby be made up of the baby cells? I mean, this is the baby. Let me ask you to remember something. You used to have an umbilical cord. You'd say, yeah. Was that umbilical cord made up of your mother's cells or your cells? Your cells. And that's why when babies are born today, they'll sometimes ask, do you want to cryogenically freeze the umbilical cord? Because it contains what are known as embryonic stem cells from your baby. Has anybody ever heard of this? So that's made up of the baby cells, that umbilical cord, as well as the sacs that surround the baby. So in this newer method, which is considered safer than amniocentesis and can be performed at an earlier stage in pregnancy, they snip off a tiny few cells from the outer chorionic sac, called chorionic villus sampling. So whether they use this amniocentesis method or CVS method, they take the cells and they can do various tests, including a karyotype. What did we say a karyotype is? A chromosome analysis, an analysis of the nucleus. Now, what would it look like if somebody has Down syndrome? There's an extra number 21 autosome in every cell of that person's body. You'd say, well, I don't get what that means. Let's look back on page M3. On M3, if we did a karyotype, a chromosome analysis, what we would find is right where it says number 21, instead of finding 2, a pair of them, we'd have 3. There's an extra one. Now that's almost surprising that that would be a problem, because our first thought is, gee, you've got all the chromosomes you're supposed to have, and you got one extra one, even to boot. That should even be better. But the way the human body works, the way all life works, is it needs the right amount. Anything more than normal or less than normal is not normal. When we talk about your blood pressure, we want a normal blood pressure. We don't want it higher than normal or lower than normal. When we talk about your thyroxine hormone level, we don't want you to have hyperthyroidism. We don't want you to have hypothyroidism. We want you to have the right level. That's the way your body works best. That's considered, quote, normal. When you don't have what's normal, it's abnormal, and things don't work right. So actually, either missing a chromosome or having an extra one is a chromosome anomaly, an abnormal count. This creates, having an extra autosome, number 21, is responsible for the most common of these chromosome anomaly disorders. This one is called Down syndrome. There are a whole bunch of these, because you might ask, well, could you have an extra number 22 or an extra number 14? Yes, yes. Could you be missing a number 16 or missing a number 3? Yes. Anything you could imagine that could go wrong, unfortunately, does go wrong, but most of these are extremely rare. But Down syndrome is the most common of these for whatever reasons. So, in terms of back on page M2, so people with these chromosome anomalies almost always have some degree of mental retardation, almost without exception, mental retardation to varying degrees. And there's usually other physical abnormalities as well. In the case of Down syndrome, they're characterized by having a protruding tongue and a short, stocky build, straight hair, and so on. And there's usually other problems with vision and the heart, and so on. It just doesn't work right. Unfortunately, there's nothing that can be done in terms of once a baby is born, because the defect is in every single cell of the body. Since the zygote started out with the wrong number of chromosomes, every cell that developed from the zygote has the wrong number. Now, in terms of why did this happen? Why? Let's go back to M3. Why was the egg or the sperm, why did it have the wrong number of chromosomes? As I said, the process of making sex cells, which occurs in our reproductive organs, is called meiosis. And sometimes it just doesn't work right. The vast majority of times, it works perfectly. In fact, I'm always surprised that it works as well as it does. All right? Because this is all complicated stuff. And the fact that so few errors occur, that's what impresses me. But occasionally, errors occur in meiosis, errors occur in mitosis. Yes?
Why when women get older?
We're going to explain that right now. So, even though errors occur in the forming of the egg and the forming of the sperm, both, we usually assume the real problem was the egg and not the sperm. Now, we're not placing blame. Nobody is to blame. It just happens. Some of the sperm have the wrong number of chromosomes. Some of the eggs have the wrong number of chromosomes. But the reason why we assume the problem originated with the egg is for two reasons. Reason number one, when a guy ejaculates sperm, he ejaculates hundreds of millions at once. A small percent of them have the wrong number of chromosomes. But it's not likely that out of millions, the ones that are the deformed sperm that have the wrong number, it's not likely they're going to be the champion swimmer and they're going to be the first one to unite with that egg. So there's a bit of competition among the sperm as they swim through the woman's body towards the egg. In contrast, a woman normally releases one egg each month. Just one. There's no, quote, competition. So that egg, if it happens to be one that doesn't have the right number of chromosomes, then it's going to be fertilized if there's intercourse, and it results in a baby with the wrong number of chromosomes in every cell. So unlike sperm where there's millions being released at once, in a woman there's just one egg released at a time. There's another difference. We know that guys, because they produce sperm every day of their life, they constantly make new, quote, fresh sperm every day. A woman only releases one egg each month. How many eggs would that be a year? 12. In the course of a reproductive lifetime of 30 years, she will only release 400 eggs. You can calculate it yourself. Just multiply 12 times 30 and see how many you get. So it's about 400 eggs. So in fact, women don't produce new fresh eggs every day. They don't need that many. The eggs, all the eggs that a woman will ever ovulate, she's born with. So her eggs are as old as she is. So over the course of our lifetime, we're exposed to things in our environment, chemicals in our food, chemicals in our air that we breathe, we're exposed to x-rays and so on, and this can affect the eggs that are in a woman's ovaries. Women do have to be more protective of their bodies than guys do. It may not be fair, but it's still nevertheless the truth. And so if guys are stupid, and they are, and they do dangerous things with their bodies, they might still make it, in spite of these stupid things. But women have to be protective not only about their own body, but the eggs they have, and also only a woman can get pregnant and carry a baby inside them. And they have to be very careful when that little baby is growing in them, because it's more sensitive to things in the environment than an adult person. So for the two reasons that I've given,
You'd say, what were those two?
guy releases lots of sperm at once, woman normally one egg at a time. Secondly, the sperm are really just, new sperm are made all the time, fresh daily, as it were, but a woman's eggs are as old as she is. So for those two reasons, the problem usually is assumed to originate in the egg, in most cases. So, on page M6, there's a graph at the bottom. And on this graph, it's basically showing the probability of having a child with Down syndrome. And the major factor seems to be the age of the mother. So here it's showing on the x-axis the age of the mother. Is she 20 years old? Is she 30 years old? Is she 40 years old? And here it's showing the number of children born with Down syndrome. And you'll notice what the graph line looks like. That it's pretty low probability of having a child with Down syndrome, but it really starts to increase after the age of 40. Now I don't want to overstate this because a lot of stuff, I'm sure some of you have heard what I've said. I'm saying now, you've heard it before. I don't want to overstate it. The probability of a woman who's 20 years old having a child with Down syndrome, and a 20-year-old who gets pregnant could have a child with Down syndrome. I know cases of that. The probability is 1 out of 1,000 births. All right, so it's not 0, but it's about 1 out of 1,000 babies born to 20-year-old mothers. At 40 years old, the probability has increased by tenfold to 1 out of 100. 1 out of 100. So for women who are 40 years of age, and their eggs are 40 years old, they're as old as she is, so the probability of having an egg with the wrong number of chromosomes now has increased from 1 out of 1,000 to 1 out of 100. Statistically, that's a tenfold increase. But I want to, again, statistics are a funny thing. And let me express it a different way that I think will help make you feel, quote, better. The probability of having a child with Down syndrome for women who are 40 is 1 out of 100. What does that mean? That means 99 out of 100 babies are fine. If somebody told you that you had a 99% chance of winning the lottery, would you buy the lottery ticket? Yeah. Of course you would. There's only a 1 out of 100, 1% chance you won't win. People are buying lottery tickets when there's a 1 out of a million chance. Here you'd have a 99% chance of a healthy baby. But we can't deny that the probability has increased. And it does get higher even with a later age. It still doesn't mean the majority of babies have the wrong number of chromosomes, but the probability is increasing. Have we answered that? All right. So we have talked about chromosome anomalies. I want to now look at page M5. On M5, so this says
Human chromosomes.
all right? And what it's showing diagrammatically, schematically, is it's showing these chromosomes. We have learned that in every body cell, there are 22 pairs of autosomes and 1 pair of sex chromosomes. Right? We've said that. Page M1 and on page M3.
You'd say, like, should we know that?
100% you should know that. 1 pair, so 22 pairs of autosomes, 1 pair of sex chromosomes in every human body cell, somatic cell. So this is showing, this is labeled number 1. That's autosome number 1, autosome number 2, autosome number 3, 4, 5, 6, autosome 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Now we normally have a pair of each of these in every cell. One of them could be traced back to the sperm from the father, and the other could be traced back to the egg from the mother. You have a pair of each. We also have a pair of sex chromosomes. If you're a female, you have 2 X's. If you're a male, you have 1 X and 1 Y. Now, as many of you probably know, over the last 35 or 40 years, there was an international effort led by the United States to map every single gene on the chromosomes, to identify what the genetic information is on these chromosomes made up of DNA. That was known as the Human Genome Project. Essentially today, we believe we know all of the genes that are on all of our chromosomes, the autosomes and the sex chromosomes. This chart only identifies some of the genes. Normally on each chromosome, there are 2,000 genes or genetic instructions. What is a gene? We wrote right here, each gene is an instruction for making 1 protein. And there's about 2,000 genes on each chromosome. 2,000 genes on each chromosome. Each gene is an instruction for making 1 protein. So these are really instructions for making proteins in our body. Obviously, there's another type of problem we can have. Not only can we have chromosome anomalies. What's that? That was the wrong number of chromosomes. We can have a defect in a gene. That's not the wrong number of chromosomes. It means we got the right number of chromosomes, but one of these genes could be defective. And if these genes are defective, which is called a genetic disease, that means what a genetic disease is an inability to make a particular protein in our body. Let's try to give you a little bit of a sense of this. You'll notice, and I'm not asking you to memorize which genes are on which chromosomes. Let's just look at a few. So on autosome number 2 is a gene for making antibody proteins. Did we ever learn that antibodies are proteins? The answer is page D5. On D5, we were learning about the importance of proteins, and we mentioned that some proteins act as enzymes, and some act as structural proteins. Some of them act as hormones. Antibodies are proteins made by lymphocyte white blood cells. Page D5, in case you've forgotten. Also, have we ever talked about a protein hormone called glucagon? Yes, you were tested on the last exam. Glucagon is a protein hormone, like insulin, that affects our blood sugar level. It actually raises it. So if somebody had defects in these genes, they might not be able to make antibody proteins like normal. They might not be able to make glucagon like normal. Let's just look at a few others. Let's look at autosome 11. On autosome 11, among the 2,000 genes that are on this autosome, this picture doesn't show all 2,000, but it includes a gene for hemoglobin. If somebody his hemoglobin's a protein, we learned it on the bottom of D5. That's where we talked about most of these proteins originally. So if somebody had a defect in that gene, a genetic disease, they wouldn't be able to make normal hemoglobin. You know what that genetic disease is called? Sickle cell anemia. And we learned about that on the bottom of D5 and the top of D6. It is an inability to make a normal hemoglobin protein so that the red blood cells are misshapen and cannot transport oxygen like a normal red blood cell can. You'll notice that also on autosome 11 is the gene for making the protein hormone insulin. So if somebody had a defect in that gene, a genetic disease, that would be called diabetes, an inability to make normal insulin. You'll notice there's a gene on autosome 11 for making a protein called melanin. Melanin is the brown-colored protein in our skin that gives us pigmentation. Its purpose is to shield us, to protect us from harmful ultraviolet rays from the sun. If somebody had a defect in that gene, they wouldn't be able to make that melanin protein at all. They would be called an albino. Somebody, and I'll just write the word, albino is somebody who cannot make any melanin protein. They are not just, quote, white in color. They have no pigmentation. They have white hair and pink eyes. They have to be very careful of harmful ultraviolet rays from the sun. They have no shielding or protection. These are genetic diseases. They are defects in the genes. Now, so we're just mentioning some of these. Obviously, if we talked about every single gene, which we're not, you would understand not only what all these genetic diseases are, some are more common than others, and what these different proteins do in the body. I want to draw your attention, though, to two genes where if there's a defect in them, there's an increased risk of cancer. So they're on autosome 17. There is a gene called the p53 gene. When there's a defect in that gene, the person is at much greater risk of developing cancer of their large intestine, also known as colon cancer. So colon cancer, colorectal cancer, And colon cancer is the second leading cause of cancer death. The number one cause of cancer death is lung cancer, mostly from smoking. But this risk of colon cancer seems to be related to this p53 gene. Now, another one I want to draw your attention to is on autosome 13, and I'm not asking you to remember which autosome. I just want you to understand conceptually what genes are. They are instructions for making proteins, and what a genetic disease is. A genetic disease is a defect in that gene. There is a gene on autosome 13 called the BRCA gene, BRCA. A defect in that gene dramatically increases the risk of breast cancer. A defect in that gene. So some of you may have even heard stories of women where their mother has died from breast cancer, their sister has died from breast cancer, and that's because they have inherited this defect in this BRCA gene. If they find that that woman also has inherited that defect, some women have gone to the extreme measure of even having a mastectomy before they develop breast cancers. Have you ever heard of that? That's because they know they've inherited that defective gene, and it's only a matter of time before they do develop breast cancer. So we see that our genes are instructions for making proteins. Not only determine whether we can make insulin, make melanin, make hemoglobin, make a glucagon, make antibodies. And if we can't make these proteins, these are called genetic diseases. Sickle cell anemia, diabetes, albinism, being an albino, and even cancer has a genetic basis to it. So that's why there's so much interest in analyzing and learning about genetics.